everyone, and welcome to our Ask Me Anything session on immunotherapy. I'm thrilled to have you all with us today, and as the name suggests, this session is your unique opportunity to ask our distinguished guest absolutely anything about their work, research, or innovations in the field. But before we dive in, there are a few important details. So this is, of course, a live session, and we are excited to actually see so many of you joining already. And many of you have actually submitted your questions when you registered. But if you haven't, don't worry, because there is obviously still a chance for you to add your questions. All you have to do is click into the box on the right-hand side of your screen and type your question in and click send. Our team in the virtual backstage will be ensuring that all questions are appropriate, so please do keep them relevant. And we'll do our best to address as many questions as possible before the session ends. Also, be sure to check out the literature section on the platform for some fantastic resources available for free. If you do encounter any technical issues, you can always click the chat icon in the bottom right corner and our team will assist you. So once again, welcome to our live Ask Me Anything session. And without further ado, it's my great pleasure to introduce our expert for the day, Professor Paul Moss. Paul, how are you?
Very well, thank you. Yeah, lovely to be with you, Lucy. Thank you.
Wonderful. Perfect. So one of the first questions we always ask is, I wonder if you could give our viewers a bit of a brief introduction about yourself and your work.
Yeah, so I'm a hematologist and look after patients with hematology problems and hemato-oncology problems. But also for quite a while now, I've been researching into immunology. So I'm very much in the area of understanding how the immune system fights cancer and also things like viruses and so forth. And so throughout my time, I've been working on immunology. I helped to develop a new form of cell therapy for patients with viral infection after transplantation. And I have a research group that studies mechanisms of immune response to cancer and immunotherapy. So I've managed to combine a career in clinical practice with research, and I've seen a lot of change and development in the last few decades.
Mm-hmm. What would you say inspired you to kind of focus on that intersection of immunology, cancer, and transplantations in your research?
Yeah, it's often role models, isn't it? And that's something I remember now when trying to teach younger people and inspire them. And when I was at university, actually, I was very interested in immunology. Immunology was a strength of the local university. And so I was fascinated. It was a developing science at the time. Would we, things like T cells that we talk about right now, we didn't know whether there was one receptor or two receptors. It was very early days. So I was fascinated then. And then I went in to study clinical medicine. And it was clear to me, really, that the immune system was going to be incredibly important, understanding the immune system response to cancer. So I was really quite fortunate to get into the field really before it exploded, actually, many years later.
Mm-hmm. Oh, absolutely. So, Paul, we will be having a wide range of questions coming in on this live session. We have actually already had one just just a few moments ago from someone named Mark who said, has any subset of COVID-19, the viral study, been shown as a viral taxid pan-reptile genetic comparison?
That's an interesting question. So that's a COVID specific immunity,
Yes.
not a cancer specific.
Mm-hmm.
Any of them been seen as a what? Run that past me, Lucy.
So, has any subset of the nCoV-19 viral study been shown as a viral taxid pan-reptile genetic comparison?
That's a tough question for me. I don't really necessarily, but we're talking about mpox response. So
Mm-hmm.
that's the current pandemic, the current infection that's of concern.
Mm-hmm.
All I would say about that is, yes, it's a concerning virus. But again, while we're on to immunotherapy, what is quite encouraging is that the vaccines against what were used for smallpox, actually, the vaccinia vaccines and the NVA vaccines that have been derived from them are cross-protective against mpox. So in a way, mpox is a new infection of great concern globally, but we are supported by the great investments we've made in vaccination over the last few decades. We do have a vaccine that's quite good. The challenge is to make enough of that and to deliver it to the risk groups.
Mm-hmm. No, absolutely. We've also had another question come in actually off the back of this one, which is from someone called Laura who said, how is immunotherapy being applied in the treatment of viral infections like HIV, for example?
Wow, that's a great question. Thank you, Laura. So immunotherapy is obviously using this immune system to fight disease. And you're talking about HIV. Interestingly, again, I don't want to reminisce too much tonight, but I was a hematologist working with patients with hemophilia, and we lived through the time of HIV, actually. And I did a lot of research on HIV at the time. We were looking at the T cell immune response to the virus.
Hmm.
And also, of course, the antibody response is very strong. Now, what we know really is that decades later, we've not really been able to, as a community, develop a very good vaccine against HIV. But we're still trying to use the immune system. Would you believe, this is really rather fascinating, Lucy, antibodies are now being used in the fight against HIV, because decades ago, we thought antibodies were going to be useless. The virus was going to evade them.
Hmm.
But interestingly, some people who have HIV infection over many years, they slowly develop wonderful antibodies that can neutralize the virus. And what scientists have done using modern DNA technology is they've cloned these antibodies from these rare patients, and they're starting to use them as injectable therapy every 6 months in patients with HIV. It's a very topical area of
Hmm.
HIV research. So it's a great question, and I can tell you that right now the immune system is being used to treat HIV.
Oh, absolutely. Let's have a look at some of the other questions that have come in. We are having quite a few questions come in the live chat, so everyone do make sure to pop your questions in there, and we will be getting through as many as possible in the time that we've got available. A really nice one has actually just come in that said, What are some of the most rewarding projects you've worked on in your career so far?"
Well, what a lovely question. I'm going to tell you one story, which I don't want to make it anecdotal, but there is science in this, which is again coming back to my work on HIV. And so we were very interested, Lucy, to try and understand how large, how strong the immune response to HIV was at that time, because we knew that patients were not clearing the infection. And so I was lucky enough to work with a colleague in America from Stanford, that famous place that we hear so
Hmm.
much now. And they had these little reagents, which they called tetramers, which were little bits of virus peptide wrapped around an HLA molecule, which is how T cells see it. And they wanted to see if these could identify T cells against HIV. So without making it too complex, basically you would take a blood sample from someone who was carrying an HIV infection, you drop these reagents into the blood and they would mix up, and then you would run an assay, a flow cytometer assay, and see if you could count T cells fighting the viruses. And we were the first people in the world to use that tetramer. And I remember taking blood from these patients and seeing that. And it really changed immunology, because those reagents now can be used to measure T cells against flu or EBV or indeed cancer. And that paper, I think, has been cited over 4,500 times. It was a really exciting time for me.
Oh, that sounds fantastic. Thank you so much for sharing that kind of story with us.
Yeah.
Um, Another one that's just come in is kind of on the cancer sort of applications in cancer theme. Are there any particular cancers that you find are more resistant to immunotherapies?
Yes, very much so. And I think these are the so-called cancers of unmet need. I think the one that stands out, one in particular, is pancreatic cancer, of course, which is becoming a real challenge in oncology because it's sort of fourth or fifth incidence rate, but the outcome is very poor. Perhaps
Hmm.
I saw recently 12% 5-year survival. And so it's becoming a major cause of cancer death, predicted perhaps by 2030 to be second. So there's something about pancreatic cancer that the current therapies, which we'll talk about in this hour, are working very poorly against
Hmm.
pancreatic cancer. And so it really epitomizes some of the challenges and maybe some of the mechanisms behind that we'll talk about later.
No, absolutely. So another question. As I said, we're going to be jumping all over the place here, Paul. How is AI being used to advance immunotherapy research?
Well, I mean, I think artificial intelligence is going to transform science in many ways. And so we're only just beginning to see it. I, th- I think where we're seeing it now currently is in data analysis. You know, when I went into science, probably most people went to a bench and did experiments, right? But now only a proportion of people do that. Many of them want to go to a computer and analyze data, of course, and that's become incredibly important with the computing power that we have. And of course, you can analyze the data that comes out of a local lab, or you can actually get data from other labs around the world and reanalyze that. And I think this is where we are starting to see AI being very important. I mean, one of the questions in immunotherapy, for example, is can you predict how well a patient will respond?
Yes.
And so AI using that deep learning technology, if you feed it enough information, if you've got those data sets, you can start to predict, you know, perhaps which treatment a patient would benefit from.
Hmm.
We'll see it obviously start to come in other areas of our work as well, where they'll start to write our papers or, you know, review our papers and
Yeah.
so forth. So there's plenty of changes I think I will see.
Hmm. No, absolutely. Someone here is actually going back to following up on the HIV and antibody question. So I think we should maybe answer this one. They're asking, how come antibodies are only developed in rarer cases?
What a great question. I'm so delighted somebody asked this, because I was at a meeting online, and the world expert was talking about this, from the Rockefeller in New York. Um, and what's fascinating is that initially, if we were to have a vaccine tonight, our antibodies start to make an immune response. Our B cells make an antibody to this vaccine, and they try their best to make the best fit, like a lock and a key. But what happens with our immune system is that over the next few weeks and months and years, if you have a chronic virus such as HIV, which still lives around, your immune system doesn't rest. And so, what it does is it starts to change. It literally mutates the DNA of that antibody producing B cell, and it starts to make all sorts of different shapes. And what's remarkable is in these patients who develop these very strong antibodies, it can take years for this to happen. Sometimes, for those aficionados on the call who understand an antibody structure, almost every amino acid in the antibody shape has mutated over the years. And so it's evolution, it's natural selection, and you're seeing just those few patients where the immune system has managed to overcome the challenge. It's remarkable biology.
Absolutely incredible. Another person here, actually, this is a really great question. Do you see any potential for immunotherapy to be used in diseases such as Alzheimer's or Parkinson's?
There's almost no area of clinical medicine now where people aren't trying to bring in immunology and use the power of the immune system. Obviously, I work on the immune system, and I think we underestimate its extraordinary complexity and power. I mean, let's take the brain, for instance, which is sublime, right?
Hmm.
But that's because it's something that we can see, it's solid, whereas the immune system is all over the body, it's diffuse, but it's nonetheless equally remarkable. In fact, when you think about the evolution of humans, I mean, the sort of organ systems that have been under the greatest selective pressure are things like the brain for intelligence and also the immune system, because our pathogen challenge is relentless and pathogens are always changing. So the immune system is changing and evolving all the time to get stronger. So in answer to the question, why can't the immune system be involved in many diseases, including, as you say, neurodegenerative diseases? And you will know, of course, that in Alzheimer's you see these things like plaques,
Yeah.
um, entangles in the brain, and indeed the therapies for Alzheimer's, which are currently being addressed, are
Hmm.
antibodies which bind to the amyloid in those plaques. So, yes, we are starting to see immunotherapy used in those conditions.
Nice. Um, One of our online questions here has asked, how can immunotherapy promote vascularization when transplanting a lab-grown organ? And can it enhance transplantation by reducing the chances of infection or organ rejection?
Okay, so now we're switching, to transplantation, right?
We're all over the shop.
Yeah, yeah. Transplantation, again, is a huge area of medical practice. I'm in Birmingham tonight, which is, I believe, the biggest transplant centre in Europe. We do a lot of transplants for kidneys, for liver, for bone marrow, and so forth. And of course, the immune system is key here because with those big organ transplants like kidneys and livers, the problem is the immune system recognizes them as foreign, and those patients have to be on immune suppression for a very long time to dampen that down. So that's where we're seeing the immune system obviously causing trouble um, in terms of rejection. And despite decades of progress, we haven't really yet managed to find out how to get specific immune tolerance to those organs. The question was related specifically to the vascularization.
Hmm.
That's a tricky one. You know, how, when we talk about vascular, we're talking about blood vessels, right?
Hmm.
Blood vessel growth is an important determinant of things such as cancer growth, transplant survival, and so forth. And yeah, they are influenced by inflammation. Typically, inflammation causes increased vascularization. That's why inflammation is red and hot and so forth. But in terms of its specific role in transplant rejection, I couldn't really say any more than that.
That's perfect. So we have actually, I know just a little while ago you mentioned Mpox, and there's been a little bit of chat about it in the chat box. One of the questions that's kind of coming up is, could immunotherapy be used to boost immunity in people who have recovered from Mpox but are at risk of a reinfection?
Yeah, I mean, the greatest protection against reinfection for most infections is the first infection, right? I mean, that's
Hmm.
one of the beauties of our immune system is memory. And many of the diseases that we know about, such as measles being the classic one, it has tremendous protection once you've had it once. So Mpox is a relatively, well, I wouldn't say it's a new infection. It's been around in small cases for a long time, but it's newly emerging. And some of these new subtypes of Mpox are certainly recent and more dangerous. My prediction would be that recovery from Mpox would give you strong immune protection against reinfection, but don't quote me on that because I really don't know the clinical data on that. But I would have thought the immune memory might be quite strong. I say that because I know that the vaccines
Hmm.
against pox viruses, and we're talking here about smallpox, which of course is eradicated. The vaccines were very good there, and I would hope that Mpox might give you good protection against reinfection.
Hmm. No, that's perfect. And still on the kind of like Mpox theme that we've got here, another person has just said, for patients with a more severe or prolonged Mpox infection, are there specific biomarkers that could guide personalized immunotherapy treatment?
That brings us on to the important question of biomarkers. I would say in that situation, it's more likely to be the clinical status, isn't it?
Hmm.
It's the duration of the vesicles, the swellings, the temperature, and so forth. Um, Rather than a specific biomarker, I would suggest.
Hmm.
Um, yeah, I think
No
so.
problem. Lovely. Um, a really nice one. What tools do you think are lacking that would aid in immunotherapy research?
Yeah, we've you know, it's a truism and often stated that science progresses very strongly through technological innovation. And that has been true. I've seen that happen. Within immunology, we've had techniques such as flow cytometry. I don't know if you've heard
Mhm.
about that. We're seeing remarkable advances now in what's called spatial biology, which is looking
Mhm.
at tissues. So you can get a slide. I think we all know what a patient slide is that we used to look at under the
Mhm.
microscope. And now you can start to look at every RNA molecule, many, many protein molecules in those systems. And you can start to understand the architecture of the immune system in those slides. So that's another example where we're seeing remarkable progress. Um, the other area, which I think we mustn't underestimate, and I've touched on this, is computing. You know,
Yes.
the ability to cope with the amount of information and data that we are getting is breathtaking and can only be done by computation. So when I was doing my PhD, we were sequencing immune receptors, T cell receptors, and literally, typing them on a screen, a single sequence. Now, of course, you can run millions, and within minutes. So that's been incredible. The question, of course, was, what are we lacking?
Mhm.
Um, I think the ability to try new reagents in a great culture system in the laboratory would be super. And we need to do that in a human based system. So people are starting to do things like taking sections or slices of a tumor and keeping them alive, and then able to put their immunotherapy in and see how well it responds and so forth. So those are the sort of technologies, perhaps linked with AI and rapid
Yeah.
throughput, where you might start to develop new therapies.
Could be pretty interesting. Um, Another one, and I'm so sorry, Paul, we're really testing you here by throwing you all around the corners of immunotherapy. Someone has just said immunotherapies are costly due to large doses and regular administration for chronic diseases. How do you think we could make immunotherapy more affordable, and how far away are we from cheaper manufacture?
Yeah, super question. Yeah, I think forefront technologies are always, quite pricey, but once they've proven their value, then the price starts to come down. Remember, as a technology, do you remember how much it cost to sequence the first human genome?
Rather a lot.
Tens or hundreds of millions of pounds. And I
Hmm.
remember it being announced when one sequence was sequenced. I mean, now, Lucy, you and I could have our genome sequenced by the end of this evening,
Hmm.
very, very cheaply. So, that's one area where we've seen tremendous progress. Now, some of the current technologies, such as cell therapy and the checkpoint antibodies that we know so much about, they are pricey at the moment, but they are used because they're cost effective in terms of the life years that they save, the amount of years of life that they save. No regulators probably would allow them if they didn't feel they were cost effective. And over time, we will see their costs come down as we obviously factors such as generic competition, new reagents, and so forth. So it's a very important question,
Hmm.
but I think that where we see tremendous clinical success, we will see cost effectiveness and so forth. Yeah.
Hmm. No, that's brilliant. Um, Michelle here has asked, can molecular imprinting with nanoparticles of the FAB region of antibodies be the next step in the use of immunology to develop targeted diagnostics and therapeutics?
That's a tough question, Michelle. I think you might be doing a PhD on nanoparticles, which I'm not. So, yeah, I mean, I think nanoparticles, the word speaks for itself, doesn't it? Very, very small particles. Will they be able to chemically mimic antibody structures? Maybe that's the sort of question that Michelle's asking there. And, I tantalized you about HIV
Hmm.
patients, some of whom develop over many years very rare subtypes of antibodies. It's possible that new technologies such as nanostructures and so forth might be able to create those in the lab in very rapid time. We talked about artificial intelligence, and just to remind you that one area we're seeing it is working out the structure of proteins. So the classic way to understand a structure, let's say my hand is hemoglobin, was to make a crystal structure. So you had crystals of hemoglobin, and then you'd shine X-rays at it, right? And X-ray crystallography, very slow, very expensive. Of course, what now AI can do is it just looks at the sequence of that hemoglobin protein and it says, I think it's going to fold like that. Yeah.
Hmm.
And it predicts that. That's called AlphaFold. And that was done by artificial intelligence. So maybe by predicting structures, we can predict a drug. And that's where nanotechnology may help.
Very nice answer. Um, I think you'll like this next one. Someone here is asking, can you talk on the COVID-19 National Core Studies program you're part of?
Yes, yes, I did. I did run that thing, and so the story behind that, of course, was I used to work for the Medical Research Council, the government science body on immunology, and
Hmm.
then when, of course, the COVID-19 struck March, April, the lockdown 2020.
Hmm.
I really just wanted to try and respond and help to do something about understanding it. Of course, immunology was always really going to be the key to the long-term control of the disease. And so what I was able to do was to bring a group of 20 universities together. And I talked to them all about what they were doing and what they were hoping to do. And I wrote a grant, and we managed to get it funded. And then we worked as a consortium. And I mention that because it's not a way that typically scientists often work. I'm not
Yeah.
saying they never do, but there is this challenge in science, which is it's been a little bit typically competitive, that you
Yes.
write a grant, your colleagues review it, they may or may not fund it. You write a paper, they read it, they may or may not publish it. And so typically in the early days of science, it rewarded lone working and aggressive competition. But obviously in COVID that changed, and it was lovely to work together as a consortium. And the other thing we did in that consortium, as you might expect, was bringing patients in and so forth. Modern,
Hmm.
very important area of modern science. And so yes, we learned a lot and managed to achieve some very good work in that time.
I think that's a lovely answer. I thought you'd like that question. Let's have a look at the other questions that are coming in. So, of course, this is a live session. We are just about halfway through. I can't believe it. But do make sure to pop your questions into the box on the right-hand side of your screen and click send, and we'll answer as many as possible in the half an hour that we've got remaining. So let's have a little look at what we've got here. Let's go for... Eloise has asked, how are clinical trials shaping the future of immunotherapy?
Wow. I was just looking. There was a remarkable paper this week talking about the treatment of metastatic melanoma. So you'll know melanoma, skin tumor, very nasty condition, certainly was in the past. And melanoma is really quite fascinating because it's always been the sort of tumor which seems as if the immune system has been trying to fight it. Occasionally, you would see people who regressed, who seemed the lesion got better. So there was something going on. And perhaps because of that, it has proved very, very responsive to immune therapy. And so there's a couple of types of checkpoint inhibitor, these antibodies that unleash immune responses. And typically they were used separately. You'd use one or the other. But there's a very important trial started quite a long time ago where they combined them. And it was led by a colleague friend at the Marsden, James Larkin. And they just published their 10-year results. And it's remarkable that those people who were in progression-free survival, well, actually at 10 years, most people had not progressed if they
Mhm.
had the two together. And so it's a fascinating result. And I was just looking at the New England Journal of Medicine where it's published. And if you just look at the front page, it's quite remarkable. There are 4 or 5 immunotherapy clinical trials. So in answer to your question, clinical trials are essential. And, they're a very dynamic area of medical research. In the past, you might think a clinical trial was drug A versus drug B, and we'll just give 500 people each, and then 10 years later, we'll see. And that still has a place. But, now people are much more dynamic. They might set several things going, and then they'll see which is responding, then they'll move them around. And they're looking to get quicker, more cost-effective answers to the clinical trials. And, again, going back to HIV, as you know, I mean, people were not prepared to accept that clinical trials were going to be too slow or too, and that their lives depended on it. So,
Mhm.
in answer to the question, clinical trials are essential. They're a very dynamic and interesting area of medical research, and
Mhm.
I'd encourage people to get involved in.
Absolutely. I've been scrolling up in some of the questions here, and I'm sorry that I missed this when we were talking about pancreatic cancer. But I thought I'd come back to it because it is a pretty good one. Someone here has asked, pancreatic cancer is often diagnosed at a later stage. Do you think there's a role for immunotherapy in the earlier stages of the disease, or even in adjuvant settings, in those patients who are most likely to benefit?
Yeah, very much. What a great question that is. I think immunotherapies, they're great. They can be great, but they don't always work, as we all know.
Hmm.
There is a tendency to think they're particularly good when the tumor is not overwhelming. Tumors are very immune suppressive. They would not have developed if they couldn't fight off the tumor, the immune system. So the idea of using immune therapy when the tumor is in a modest early stage is, I agree, very attractive. So that brings us to, how do we detect cancer early? This is probably one of the biggest areas of medicine right now, and
Yeah.
possibly the biggest area of cancer right now. Um, we've got to detect it early, and you might know there are some remarkable things going on. Using the more old-fashioned things, you can do tests of stool samples to look for blood from colon cancer. People who smoke, there's studies now doing scans, CT scans for early detection.
Hmm.
But what I would really pick out, and it's a fascinating area, is using blood tests to detect cancer. And there's some very, very big international trials now. So if you can do a blood test and if you can detect pancreatic cancer early, then I agree with the questioner. That might be a particularly interesting time to try your immunotherapy.
I really like that answer. Um, there's one here that's talking more on cost of treatments. So it's Rob that's asked, the cost of personalized immunotherapy is prohibitive for many patients. As immunotherapies become more complex and individualized, how can the field address issues of accessibility and equity in treatment?
Yeah, great question, Ralph. And you talk about personalized, individualized. Yeah. On the one hand, in many ways, that's something that we've been encouraged to think about in medicine, so that not everybody gets put into a basket and gets the same therapies. If we can make treatments more personalized, more individualized, then perhaps it would be better for individual patients. But what you've raised is the nature, how expensive that can be.
Mhm.
Let me give you one example, which is very live right now and very exciting, which is vaccination. Yeah, cancer vaccines are an interesting topic. And so, what is happening? A couple of the big companies who did so brilliantly in COVID, BioNTech, Moderna, they showed the power of mRNA vaccines. And so, what now can happen is that a patient can, if they develop cancer, A biopsy can be taken. The cancer genome, as we talked about, Lucy, earlier, can be sequenced.
Mhm.
You can find the mutations, and then you predict what vaccine protein you need to put into your mRNA vaccine. You might say, this patient has a mutation in SMAD protein or something like that. And then in your mRNA, you insert that mutation as an immunogen. And then you give that vaccine to that patient. It's entirely personalized based on their genome sequence. Remarkable, but very expensive, right? And
Mhm.
so, that is the thing, that the challenge. Now, of course, the companies would say, well, mRNA vaccines don't have to be that expensive now. You can, again, rapidly synthesize mRNA. A genome sequence doesn't have to be that expensive. So, if those work, we could see the price fall quite significantly.
Mhm.
And then on the other hand, some of our, very successful immunotherapies, like antibodies to PD-1, the famous drugs nivolumab, pembrolizumab, they can be used for everybody. They're not patient specific for a disease. So look, equity, costs, whatever, are very, very important considerations. And I think that, they're particularly important once we have something that we know is very effective. Some of these clinical trials, of course, are necessarily expensive because we're trying to prove their value.
Mhm. No, absolutely. Um, I've seen a really nice question here about the microbiome. So this person is saying the intersection of immunotherapy and microbiome research is becoming increasingly prominent, or at least they are saying so in their echo chamber. How do you think manipulating the microbiome could enhance immunotherapy outcomes? Is it something that you're seeing people addressing in this field?
Wow, we're getting really great questions. These are the core of modern medicine. And so I love this, microbiome question.
Mhm.
I went to a talk recently, and they said, look, humans are meta-organisms. We hear a lot about metadata.
Mhm.
There's a company called Meta, I think. It's
Yeah.
all about integrating all these different data sets. A meta-organism is that we have our own cells, but we have more, or at least the same number of bacterial cells. So we are harboring all these different organisms, and we couldn't, of course, survive without bacteria. So the microbiome is a very, very hot area of research, to use a cliche. I think we have to be cautious because, again, it's an evolving area of science. The microbiome will be incredibly important, but we mustn't overstate some of the early findings and overinterpret them. I think
Mhm.
the questioner talked about an echo chamber. And
Yeah.
I think people who are microbiome advocates, we just need to be careful and do these studies properly.
Mhm.
But the microbiome does regulate the immune system. People talk, some of the facts are really quite stunning. Most of our immune system is in the gut, and it is surrounding the microbiome. People talk of the microbiome as a nursery for our immune systems. Yeah. And
Mhm.
so it is integral to how our immune system works. And therefore, if you go up to a ward and inject an immunotherapy to a cancer patient, the microbiome that they hold at that time, which may have been damaged by chemotherapy and all sorts of things, could well be a critical component of how they respond.
Mhm.
And that is why there's a lot of interest in trying to find the right microbiome and trying to repair or replace it with the one that gives the optimal response. Very interesting biology.
Great response. Thank you so much, Paul. Um, Again, I'm spoilt for choice on these questions that are coming in, to be honest. Let's have a look. We've got one here from someone called David who said, would cancer immunotherapy research be benefited by more widely available slash living cryopreserved tumor tissue?
Yeah, wow. That, this is Really interesting. Yes, is their real answer. Access to tumor tissue is incredibly important. You might think it's obvious in a way, but it's not as easy as it sounds, and it can be quite expensive. You obviously need to consent the patient. A sample has to be taken by the surgeon, let's say, in an appropriate manner. And of course, the primary use of that tissue is for diagnosis. So most of it goes to the pathologist. They might want to see whether the margins around the tumor are free, and therefore they want most of it. And then the researcher is on the side saying, please, can I have some of this tissue to do my research? And so there are a number of logistical challenges in getting those tissues, but we need to try much harder as a community to get those. I agree, it can be quite expensive. We also need greater equity in terms of the diversity of the population we're serving, and take those sort of factors into account when we're collecting tissue to make sure we've curated samples and data sets from everybody.
Mhm.
But yeah, tissue collection is vital.
Massively. Um, Another question here that I think is actually quite interesting. Do you think that the current focus on immune checkpoint inhibitors has narrowed the field, potentially overlooking other avenues of immunotherapy, like enhancing antigen presentation or stimulating innate immunity?
Um, yeah, almost everything in life is grey, good and bad. Is there pros and cons? Yes, you could say checkpoints have been so great that they may have taken away some of the interest. I would counter that and say, actually, they've supported everything else, because what they've shown is the tremendous power and value, clinical and, dare I say, financial, of immunotherapy. And that has transformed the field. It was 2012, I think, was the first paper um, on PD-1 inhibition. New England Journal of Medicine, Topper Lyon was the first author. And if you consider the hundreds of billions of investment that have gone into immunotherapy since that time, all aspects, I think everything has benefited, to be honest. And remember, many, many patients don't get an adequate response to checkpoints. And this is where other therapies are very needed. Yeah.
Nice answer. Thank you very much. Another one here is saying, given the rapid advancements in immune modulating therapies, what do you think are the big questions in immunotherapy that researchers should be focusing on over the next decade to push the field forward?
Yeah. We've got to look forward. I mean, we're being very upbeat about immunotherapy, and that's exciting, and many lives have been saved. But remember that there is still huge unmet need. There's unmet need in terms of response. Not everybody is getting cured by any means from these therapies. And the other unmet need, Lucy, of course, we haven't talked about, is the toxicity of many of these treatments.
Mm-hmm.
And if you were to take an immunotherapy such as a checkpoint inhibitor, which is unleashing T cell responses against the tumor, it's no surprise that you are unleashing T cell responses against other tissues and causing inflammation.
Mm-hmm.
In that paper that I talked to you about metastatic melanoma, where we're seeing very, very good clinical responses, there are also many people who are getting toxicities because their immune system is hyperactivated.
Mm-hmm.
By the way, interestingly, those two often correlate. And so people's immune system gets activated, they can get side effects, but they also get a good anti-tumor response. So we have unmet need in terms of response, and
Mm-hmm.
we have unmet need in terms of toxicity. So those are some of the big challenges that researchers on the line, hopefully some people now doing PhDs, postdoctoral jobs, clinical trials or whatever, those are the areas where we need to work. And there is really a lot to do.
Mm-hmm. We've had a really nice question kind of come in about toxicity as you've been speaking on it.
Yeah.
This one's saying, do you believe the use of combination therapies, particularly those targeting multiple immune checkpoints or incorporating immunomodulatory agents, will inevitably increase the risk of toxicity? And how can we better manage or predict the synergistic toxicities in these cases?
I think that's a really interesting question. Synergistic combinations are being
Mm,
tried a lot. So it's typical in medicine. We use one thing and then we might get a response and we start adding other things on. When you look at the history of cancer research, interestingly, it started really with children with leukemia, where it was, invariably a fatal disease. And they started to use individual chemotherapy drugs and saw a response for a short time, but it didn't last. And then they started to use combinations, and this overcame the resistance, and they started to see real cures. So this concept of using combinations of drug is one that's embedded in
mhm,
oncology. The treatment of lymphomas with things like CHOP chemotherapy is another classic example from probably the 80s.
mhm,
And so when these chemotherapies came out, they started to combine them. Now, there are a number of challenges with that. One is, as you say, does that bring more toxicity? It can do for sure. But remember that the immune system, as well as having an activation arm, also has an inhibition arm. So it can control itself. And so if you were to be able to design combination therapies which not just accelerated the activation but gave some control as well, you might have a combination that could control toxicity.
mhm,
That's a dream, but that, of course, demands quite a lot of knowledge of how the immune system works. And it brings us back to some of the questions that we had earlier about what are the best laboratory assays, cell samples, cell systems that we can
mhm.
use to predict efficacy and toxicity. And the other thing about combinations, also relating to early questions, is the financial toxicity. Yeah,
Yes.
these
Mhm.
are very, very expensive. So combinations are tricky. We're in there already. We're going to see more, but they bring those complications.
Lovely answer. Thank you ever so much. A lot of people are actually just writing in the comments that we're having great discussion. Thank you so much for answering their questions. So just thought I'd mention that people are loving it. Someone here has said, are there generalized observations on immunotherapeutic responses linked to aging?
Yeah. No, that is interesting, isn't it? If you were to draw a graph of the risk of colorectal cancer against age, certainly if you logged it, did a logarithmic
Mm,
plot, it would be a straight line. So as we get older, we develop more cancers. Now, the obvious suggestion there, the obvious, probably one of the major reasons is that we acquire more mutation damage as we aging. We're getting cosmic rays all the time. We're getting radon from, we eat and drink mutagens. So it's
yeah.
not surprising that we get more. But for me as an immunologist, the question is, does our immune system start to fail in its surveillance of our immune system? And so, that's a valid question as well. Now, in terms of toxicity, of course, people are and efficacy, people as they get older are more frail, and therefore they can tolerate less treatment. I personally don't know the data on things like checkpoint toxicity and whether and efficacy, how that relates to age. But the whole area of immuno-oncology in older people is a big one. One thing, Lucy, if I may, is tonight we haven't really talked very much about cellular immunotherapy so much. And,
Hmm.
when I was a doctor, well, I still am, but when I was on the wards, I was looking after patients with bone marrow transplants,
Hmm.
so what we call it stem cell therapy. And, the concept of a bone marrow transplant came from actually the days of sort of atomic worries about atomic weapons and so forth.
Oh.
people were going to have their bone marrow destroyed by radiation.
Hmm.
Therefore, you could bring in a bone marrow from another person. And that's how it developed. And really, the whole concept of bone marrow transplants was people with leukemia, you could give them very, very big doses of chemotherapy, so their bone marrow would disappear, and then you could replace it. It was all about big dose of chemotherapy.
Hmm.
But then in the 80s, what became clear was that it wasn't just about that at all. It was about the immune system of the donor coming in and killing the tumor. It was immunological.
Hmm.
And we now know that the immune system plays a huge role in curing people with leukemia after a bone marrow transplant. And the reason I mention that is because there is, of course, an explosion now of cell therapies, stem cell transplantation, CAR T therapy,
Hmm.
tumor infiltrating lymphocyte therapy, or whatever. So we've talked quite a lot about antibodies and so forth, but the whole area of cellular therapy is also enormous and an area of great interest.
Hmm. Someone has actually just asked a question in the background whilst you were speaking on this too. They said, in your opinion, do you think immunotherapies will eventually replace, or would they just kind of be used more to complement transplantations? I guess in bone marrow
Yeah,
transplant.
I think they will. Yes, I mean, in everything, all of this will happen. I'm very, very confident that we will control cancer. Within, I would say, within a generation. I'd be very optimistic. I think that things are progressing very, very well.
Hmm.
We want to get rid of non-specific chemotherapy. You think of the old chemotherapies, which have saved countless lives. Don't get me wrong.
Hmm.
Sort of anti-metabolites that stop cells dividing very rapidly. They're very non-specific. And so I think what we're trying to get onto is targeted therapy. And we are starting to see diseases, certainly in hematology, which are now chemotherapy free.
Hmm.
Cancers are chemotherapy free. And so I believe that we can do that with immunotherapy. Um, so yeah, the question related also there to stem cell or bone marrow transplantation.
Hmm.
On the one hand, it's magnificent because it can cure otherwise incurable cancers. On the other hand, it is troubled by significant toxicities. And therefore, yes, we've got to try and understand why transplants work so well, but enormously reduce the risk and the toxicity. And that's an area that my own group and others are working on.
Nice. You've now kind of started a little thing about sort of graft versus host disease and everything in the comments here. Let's have one of these. So, what are your thoughts on the use of mesenchymal stem cells, MSCs, or other immunomodulatory cells in preventing graft versus host disease whilst preserving the immune reconstruction post bone marrow transplant?
Yeah. So let's just explore that question. So, yeah,
Mm,
typically with a bone marrow transplant, the old classical thing, and I'm trying to use an analogy here. So it developed really with brothers and sisters because who were tissue typed
mhm,
because they had to have the same HLA and so what happens, of course, is that you have a leukemia, and then the patient's brother or sister gives their donor immune system, and it can kill the leukemia, but it also can kill other tissues. Yeah.
mhm,
So, and that is why we see this condition called graft versus host disease. Now, again, and you would have predicted this from everything we've said tonight, people who get graft versus host disease have a better cure rate as well because
mhm,
the immune system is doing both. So, what we've really wanted in the last 20 years is to stop graft versus host disease, but allow the tumor response. We're trying to
mhm,
split these two immune responses. I don't think we've entirely succeeded. But the question there related to approaches to that. And one of them is to infuse these, what we call mesenchymal stem cells. They're
mm,
an enigmatic cell type that you typically get from bone marrow. They're a bit like fibroblast cells, but they're quite immune suppressive. And so what people have done is they, believe it or not, somebody who has severe graft versus host disease, they'll infuse into the arm these cells, and they will dampen down the immune system.
Okay.
So they've been around for quite a long time. They are intriguing cells, and that's one approach to try and separate these processes.
Hmm. Very nice. Thank you so much. We have only about 6-ish minutes left, but there are just so many good questions here. It's so difficult to choose from them all. Someone here is asking, what's the latest research in immunotherapy of vitiligo?
Vitiligo. Well, what an intriguing condition. So vitiligo is obviously the condition where people um, get very depigmentation of the skin. So where the melanin is lost. Now, that is an autoimmune condition. So that's where the immune system turns on our own body against melanocytes. We haven't really talked too much tonight about this whole double-edged sword that we want our body, our immune system all the time to be fighting our cancers and stopping cancers. But on the other hand, we don't want our immune system to be damaging our normal tissues.
Yes.
And our bodies have evolved and developed that we have, as well as immune system activation, we've got a lot of breaks, and we have to have those. Now, autoimmune disease is a huge problem. Despite all of these breaks, we're just poised that we get a lot of autoimmune disease, multiple sclerosis, rheumatoid arthritis, vitiligo, endocrine disorders. So you can see how we're just living on the edge all the time with our immune system. Now, vitiligo is an autoimmune response to the melanocytes. Melanocytes cause melanoma, right? So, you might say, would the two be related? And yes, they were, because in some of the early clinical trials of patients with melanoma, the patients that did very well did develop vitiligo because the immune system was fighting the melanocytes, it was killing the melanoma, but it also killed the healthy skin melanocytes as well. So it's a very nice question because it shows this hydra, this double-edged Janus-like complexity of tumor responses against cancer and immune responses against normal tissue.
Mm-hmm. No, thank you ever so much for answering that one. There are just some really nice questions here, Paul. Let's have a look. Someone has said, what keeps you motivated day to day?
Yeah, I mean, I've got the wonder of biology, which is remarkable, and it amazes me more every day. And also because it is a remarkable time that we're living through. We must never take granted what we're living through. We see these remarkable Kaplan-Meier survival curves where, a patient survival curve is like that. And we're seeing drugs that are moving things up like that. Now, you won't see that ever again because that now is the normal. And we're now trying to get that survival curve up to 100%. We're living through this remarkable time, but there is still unmet need. We have these tremendous tools. We have investment. I can't see why anybody would not want to get involved with this project. And it's so important and exciting.
Wonderful. I think that was a lovely way to kind of close the session because actually we're just coming to the end.
Okay.
So yeah, thank you so much for joining us today, Paul. Unfortunately, I actually can't believe I'm saying it, but the hours hours flown by.
Great.
We've reached the end of the session. It's been an absolute pleasure speaking with you. The feedback, even in the comments just now, has been fantastic. Everyone has enjoyed the discussion. So thank you ever so much for joining us today, Paul.
No, Lucy, thanks for being such a great host. And to everyone online, A, for joining us, and B, for such great questions. I couldn't have plotted them myself. Lucy and I haven't prepped this. And there were tremendous questions. We covered so much. I really enjoyed it.
Wonderful. Thank you ever so much, Paul. And before we close, I'd love to hear from you all as well. Did you enjoy the session? And if you did, please let us know by the feedback form that will be appearing on your screen very, very soon. This AMA will now be made available on demand, so do feel free to share the link with anyone who might be interested. And once again, thank you all for your time and for your participation, and we'll see you very soon.
Thank you.